Next, we have Joseph Deeb with us, speaking from STgenetics.
Good morning. Okay, well, it's always a pleasure to be back to Iowa State, although I didn't do that often since I graduated or since I left the SUS lab in 2002. Now, I hate to admit how long it's been, but you know, you can tell for how much hair I lost since then. As we say, it is main problem with hair is not about production, about distribution. So, more problems other places. But it is like an honor to be back here to just recognize Sue's lifetime achievements, lifetime contribution, not only to the poultry community, but all species worldwide, right? As evident from the variety of speaks or talks we've seen today. And today I'll give you a little bit also my take on genomics specifically, starting from chickens and ending now in the bovine side of things. I am the Vice President of Genomics with the sexing technology. Some of you might not be aware of what we do, so just one slide in a nutshell what we work on. ST started as, or continue to be, the leader of providing sex-sorted semen for bovine worldwide. We have 53 labs in 20 different countries. Our business model on the sorting semen is we build the lab, we populate the lab with the machines, and we hire the people, the technicians that run those machines or provide services for the stud that is next door, basically. They collect the stud, will collect the semen from those bulls, provide that semen to the lab, we'll be sorting, and we charge based on how many straws of semen we produce out of that batch of semen. By the way, when we are traveling with my colleagues at the airport, we try not to use too much, talk about work too much, because a lot of that is related to semen production and jump bulls and etc. I spend most of my time in this area on dairy and beef genetics and genomics. I'll give you a little bit more about that in a minute. But also we have, some might not be aware that we also have a pig sister company called IQFast, where we actually, my team is acting as the research arm from that division. We're not involved in the genomic or genetic evaluation in the porcine side of things, but we are the research arm of that group. And we also run our own genomic lab. So everything we do, so we have genetic vision since 2016, about the same time when I joined ST. We actually run our genomic testing in-house, and we provide also genomic testing for all customers worldwide. Like anyone want to genotype or sequence, we are in any species, mostly bovine and porcine at this point. It can submit samples to us, and business has been going great since then. My long journey in genomics started with these three birds lines that Sue continued to maintain. And back then, if you remember, more than 20 years ago, before any of those species have been completely sequenced, before having any like real resources on genomics, to actually do that kind of work, you need to control LD. You need to control, number of haplotype that's segregating in the population, and the only way we could do that is by creating like a resource population of an F1 and F2 populations. And I don't have a lot of pictures of when we're having fun, but this is my way of introducing my type of fun. We had a great group of people that helped in developing and testing these populations. Of course, the leadership from Dr. Sulamont and beautiful genetic variation. What else you can hope for, right? So that population, the Iowa Growth and Composition Resource Population, became my first introduction to genomics, as we started actually an effort to map QTLs or genomic variants that are correlated to those kind of traits, so growth traits. And this is not progressing. There we go. So, and we had some happy accidents in the process, right? This picture still amazes me all the time, every time I look at it, right? We have the broiler population. This is the distribution of their body weight. They're all at the same age. And these are the two inbred lines that contributed to the resource population, the F1 and the F2. And you see the distribution of their body weight at 8 weeks of age. What we expected when crossing the two extreme lines is to get like a variation that, as expected for any Mendelian segregation, you get a variance that actually covering the entire range of phenotypic variances from the highest broiler to the lowest inbred. But we got really a very truncated genetic variation here. And we went into publishing that in a very nice article in the Journal of Heredity. Again, a long time ago, but and talking about the evolution of or how the genetic contribution or the genetic variances that contributed to the performance of the broilers as we measure it today, the modern broilers. And the idea that we argued there that these animals are not there just because an accumulation of additive effects, but also because of interaction of additive by additive and an accumulation of a lot of small genes or a lot of small a lot of genes with small effects that actually accumulated over 50 years of intense selection for body weight. And that to reproduce that genetic variation in one generation on F2 was almost impossible, as we discovered. That wasn't the case for all the traits. And you see like trait like drumstick weight as percentage from body weight. We saw, you know, as expected from an F2 population, we get a big distribution of those animals. And I went into publishing my first paper in genomic selection on genomic mapping in animal genetics using the F1 population for that. And for that paper, we had only a handful of microsatellite markers in two broiler population, like sire population, half-SIP populations. And I argued that in that paper that with those hundreds of markers, we covered more than 90% of the genomic variation exists in that population, which wasn't a lie, but was given that show you the limitation of this kind of effort is you ended up with QTLs that cover half of a chromosome in the best case, right? World have changed since then. And, you know, about the same time, all the effort of sequencing and all the major species have started and completed about the same time. I worked in all those species in different capacity over the years. And of course, the world didn't, we took it very easily, right? I wouldn't, even if I commissioned like someone to actually explain how I felt at that time, I couldn't come even close to this picture because I was working in pigs at that time. And our sister company was already doing with ABS, we were doing already talking about genomic selection, right? So, and I had only a little laptop to deal with that fire. So the fire was next door. I know it's coming. I didn't have any tools to deal with it, but and the whole industry was actually feeling the same thing. If you remember, a lot of those discussions back then were around, you know, how we deal with that, how we can afford genomic selection, how we can, you know, do we phenotype? We don't phenotype. What is the best approach, genomic, you know, statistical approach to deal with the base A, base C, base B, base pi, all those things that happened, right? The missing heritability questions. You know, how many times, how many years we spent actually debating those things, but eventually things settled very quickly. And if I have to admit it, although I wasn't admitting that this fact back then, but today I do. We wouldn't be doing genomic selection today in any species if it wasn't for the dairy industry. And if I be frank with myself, here we go. And why is that? Because it's a poster child for genomic selection, right? I mean, all of those things you're familiar with, why genomic selection should work or shouldn't work. And the bovine world was actually the poster child for just hitting each limitation of traditional selection and why genomic selection fit nicely into that. But just for those that are not familiar with the dairy progeny testing cycle, if I have a bull today that I want to use it as a breeder for the next generation, I need to calculate the breeding value of that bull. And that breeding value coming from his daughters that have milking performance. And that doesn't happen until 5 years later. So I have, before genomics, I have to park this bull in my stud eating and having fun until for 5 years to decide if he's gonna be the breeder that's gonna sell semen out of that bull, or he will go to McDonald's. Today in genomics, the minute I can sample DNA out of that animal, and as we will see, I don't have to wait in a minute, but we don't have to wait until he is born to actually decide about his fate. The minute we have a DNA sample of that animal, we can accurately calculate genomic breeding value of that bull, right? And today in the structure of the dairy industry, the accuracy of genomic breeding value is at least 50% higher than what you get from traditional pedigree-based genomic selection, right? So that actually enhanced and changed all how we do business in genomics. And I put up here the breeder's equation. Of course, so this is real data from US data. The animal age here and the parent's average age. So this is what you can see in 2009 with the implementation of genomic selection. You start to see a reduction of the average age of animals used as breeders for the next generation. I don't have to wait anymore the 5 years before I have to decide about the age of that or the if I use that bull or cow as a breeder for the next generation. And this has really happened very, very quickly since the first genomic selection started in 2009. Again, that's generation interval. You reduce generation interval, you increase genetic gain, right? It's 1-to-1, and this is also true data. This is not made up data from the US dairy records, right? After 2000, since 2009, we tripled the genetic gain in this breed. And today we still publish progeny tested bulls, right? We call them proven bulls, and 3 times a year, but we also publish young genomic bulls that have 0 daughters in the industry, right? And those are the top one or segment of the top 100 bulls that are published by the Holstein Association. The yellow ones are our bulls, and you know, we are in the dairy industry, we like to say, oh, we're doing better than others. So okay, shifting gears a little bit now and to talk about the problem with the dairy biology or reproduction biology in dairy, right? I mean, the gestation period is 9 months, so basically you cannot create more than 1 animal per cow per year, right? And that's a big problem for us, because once you have the right genetics, you want to produce as many of those as possible. And the way we go about, and most decent companies do that, is when you have like a high genetic cow and high genetic bull, you collect oocytes from the cow, you do IVF, you find a bunch of surrogate mothers that you implant the embryo on them. 9 months later, you genotype all the F1s, or sorry, the F1s, the full SIPs, and you find the one that have the highest genetic value, right? And that's becoming your breeder or some of them. Okay, and we don't stop there. Well, one thing we do as well, we do a lot of cloning, some cloning, right? So when you have like a young animal that we know based on genomic value already, when he's born, we know that is going to be a really high genetic value, we can immediately start cloning out of that. And Captain is our #1 worldwide proven bull in the Holstein industry for 3 years in a row. And we have his 2 clones, Jack and John, that actually help us to produce more semen out of the same genetics. But we don't have to wait for the animal to be born to actually learn about the genetic value of that animal. Early on, as fetuses, we collect amniotic fluid from all those pregnancies. We isolate embryo cells, we genotype those embryo cells, and before the pregnancy is completed, before that come to term, we know what to expect in terms of what genetics is coming up, right? And this is important because this is the way we can manage our genetic resources, right? If I know that I'm hitting where I want to be in terms of genetic gain, I don't have to continue producing the same bull and the same cow, because those host females are very limited. But also we don't have to stop there or to do that here. We can as we produce IVF embryos, we can dissect those embryos, create a cell line out of those embryos. I don't know what's happening. So we actually create cell lines out of those embryos. And we genotype the cell line.
The communication is
I don't know what this. So.
This one.
Hit it again.
Is the system here?
It is showing well on Zoom.
Yeah, it's the system here. This thing that we do that.
Stop the clock. Stop the clock.
Now it's showing.
Okay, so as I said, we don't have to actually implant every embryo we produce. We can create cell lines of those embryos, genotype the cell line, and once we have the cell line that actually have the right genetics, we clone that, right? And then from that clone, we produce that just one specific animal that we want really to create. So imagine how fast this actually, how fantastic this tool for us to overcome a very limited, or biggest problem in dairy is that biology and reproduction problem, right? So you can do all the selection you want in a petri dish, right? Before you actually decide on what genetics. But we also don't stop there. Those females, to produce the right oocytes, typically you need a year and a half, something like that. We have also a program called GIVIT program, where we actually able to produce viable oocytes of a young female as early as 3 months old, right? So this whole program, again, I put back the breeder's equation here just to figure out how fast this genetic progress has become in dairy. And we've produced a pig out of a dairy program, basically, and halved again and again the generation interval. So we have genomics that improved the accuracy of genomic selection, right? Again, reliability or accuracy. Selection intensity is went to the roof with this program, right? We cut more than half the generation interval. So the future for the dairy industry is from application of genomics has been fantastic. Okay, I said we have our own genomic lab. We run genotyping, we produce our own chips. So to keep up with any new mutations that we discover through our research or some in the literature, etc. We produce our own chips that we use in our genomic selection for bovine and porcine. We provide services for other companies as well, or other people that want to do genomic selection or genomic testing. But we also start doing a lot of sequencing. And the way you're all familiar with this graph, basically the reduction of the cost of genotyping or sequencing over time from year to year. But, if I had to guess today is about, $500 to $800 to get like 30x whole genome sequencing in an animal. And still it's an expensive effort to do that regardless of, not in the mass production or the mass amount of sequencing we wanted for this type of research. To go around that cost of sequencing, we do scheme-seq or low-pass whole genome sequencing. And the idea is very similar to whole genome sequencing. The only big difference, instead of sequencing for 30x, we sequence for half x. And typically with our sequencing depth about 0.3x. And the idea here is that we use imputation to compensate for that lack of information that's coming from actually reading the individual nucleotide across the whole genome, right? And this is proven to be a very, very accurate method to actually to fill the gap on missing sequences. And that's also only the function of how good is your haplotype library. So today we have about, I don't know, 4,000 animals with high depth sequencing that are providing the resource to do accurate imputation. And we do that at least twice. We have 2 cycles about twice a week of sequencing that we do. The reason this started is because, remember, we still a semen company, we sell semen. So this has started because we had a problem when every now and then we have a mistake that we have the wrong bull actually ended up in mixing semen from different bulls in the same batch. And to overcome that problem, we decided to do sequencing, and we can talk about why genotyping doesn't work, but take my word for it, genotyping is not sensitive enough to identify those kind of level of contamination. So this is a trial that we run where we mixed semen from bull A and bull B in different levels, and this is showing that this is our proof of concept, the first trial I did in this experiment or this effort, and we show that even at 1% level of contamination, we can tell the difference. We can tell that semen has been contaminated or not from a semen from different bulls. But the beauty about doing this, we do that for every batch, and every bull is collected multiple times a week, and every jump, we sequence that to half X. So what happens is every jump we collect for half X, we accumulated the FASTQ files from that, and before you know it, you have high depth, really whole genome sequencing on every bull. So basically, my research program is benefiting from something that is a byproduct of a bigger problem. So I'm getting free sequencing, basically. This is just showing how many SNPs we have by chromosome. We call about 30 million SNPs on every bull we create, we sequence. We run over 100,000 reactions. So even if it is half X, but imagine like 100,000 DNA extraction, 100,000 library prep, 100,000 sequences, 100,000, reading those FASTQ files, doing the imputation, reading the mutation, creating the PCA files, all that thing, managing all that data. It has been an amazing and outstanding work that my team has been doing for the last couple of years. So we have a bunch of bulls that actually reach really high level of depth, and we continue to add as we go. Why genomics is important? Because resolution in genomics is really important, right? And I'm using this as just an example of one mutation that was published about a year ago, was discovered at Penn State University, a mutation that causing what now is called the muscle weaknesses. Basically, any animal that have a T in this position instead of C, basically unable to walk and either die or euthanized shortly after. And this is why this mutation is not tagged by any other markers nearby. By the way, we learned, despite that everyone is talking about the Holstein population as one big happy family with big haplotypes and very small population, effective population size, we found a lot of SNPs that actually don't have a tag SNP close to them using that, the usable SNPs. And that helped us a lot in fine mapping a lot of those QTLs. I don't have a slide about that, but take my word for it. There's a lot to gain from that high level of genomic information that we use. Just an example for something different, right? It's not everything we do is about Mendelian genetics. We also track some defects like monosomy in this case. This is an embryo that we produce and actually happened to be missing one of chromosome 27. We see that when you look at the heterozygosity level by each chromosome, you see that from looking at the Mendelian errors from that embryo against the 2 parents, and of course from the intensities. You could do that from genotypes of at that level when you're missing full chromosome. You can do that from the markers from the genotype level. You don't really need sequence, but we also, the same process, we tag smaller non-Mendelian defects. Switching gears here, so we not only, so the next level of doing genomics is actually providing solutions for the next generation. And that's where chromosome with matings has come to play. So big part of what my team is doing is looking into chromosome, into designing a matings program that to get you to the level where you want to be, right? And for our customers, we already published this chromosomal, what we call chromosomal mating. It's just sexy name, nothing else. But it's basically a program that is using genomics. We calculate for each mating, we calculate the genomic relationship between the animals and that you have in your pool of semen and females that are available. We look for frequency of lethal haplotype. We try to avoid the carrier to carry all those kind of things. But with the main objective is to move the population average up without affecting the variance, or if you can actually reduce the variance and managing breeding, etc. But that's nothing new here. That a lot of people do that in different versions of this. What's beautiful is this our internal program, what we call it chromosomal painting. I don't want to go too much into details here, but this is Captain. Again, Captain is #1 genomic or proven bull in the world. This is his TPI. TPI is one of the total production index, one of the indices we select for in dairy. You can see in the table that he is really ranked #1. And intuitively, we all know that 3,331 is not coming from one position in his genome. It is a lot of pieces of his genome actually contributing to that value. But when we design matings, we actually don't think about it that way. We have high TPI bull, we have TPI cow, and hoping for the best. What we try here to do is actually open that black box and look at each segment of his genome separately, right? And this is his painting, his 29 autosomal chromosomes. In blue, what you see are segments of his chromosomes that are positively contributing to his genetic value, and in red are segments in his genome that are negatively contributing to his genome, his breeding value, right? And we do that by calculating, by assigning a value for each SNP, then phasing that genome, and then assigning a value of additive value of those to the haplotype. We do sliding window of those haplotype, and that's where you get the different colors of those things. To contrast, I guess another bull that wasn't as lucky, you see a lot of red, right? I masked his identity just for his privacy. You never know, right? But so to design matings, now we do even a little bit more than just looking at his profile. So this is a different bull. Ripcord is a grandson of Captain. What we do, we also, because we know the haplotype, we phase each haplotype for each chromosome, right? And we know the hotspot for crossing over. We can actually take each chromosome and simulate gametes out of that chromosome. So that's what you see here. These are the distribution of 3,000 simulation of each chromosome, and what is the expected distribution and the mean of that distribution. So when you see like a bigger variance, this is because there's one QTL basically that is not always inherited with the same haplotype. But also you see like some chromosomes, even with the highest, this is the Ripcord is the highest genomic bull in the world today. Even with the Ripcord being that high in his genetic value, still have chromosomes that actually dragging him down, like chromosome 18, 22, and 24. And the idea here is, can I find a female that she has a similar profile as him or even average in most of the genes of the chromosome, but also compensate where he is lacking, right? And that's the level of genomic breeding that we actually start implementing. Again, the idea here is to create outliers. How many outliers I can produce from this? The flip side of genomic progress, of course, in breeding. And everything I said about genomic progress, you can actually think about it in breeding terms, right? In breeding is homozygosity, grounds of homozygosity, or identity by descent in this case, right? You can design matings that avoid animals that are homozygous for the same region, right? I don't care if he has 20% inbreeding, she has 20% inbreeding. If they have inbreeding for the different chromosomes, why I should care, right? And that's the level we look at. Here's the contrast between the top panel, just an animal that's 65% inbreeding. Actually, it's not an animal, just a cell line. I don't know who had the idea to create that thing. And an animal that have very little inbreeding in the bottom, just as a contrast. I talked a lot of some fun stuff, but honestly, a lot of what I spend it here in traditional animal breeding and genetics. Basically, we do a lot of the genomic prediction, genomic breeding value calculation, accuracy prediction, all of that. We talked about matings. We do genetic audits for all our customers. We provide that kind of service to them. We build tools actually to track progress when customers that use chromosomal mating to advance their breeding program. We give them feedback and the tools that actually allow them to gauge their progress and design their mating for the next generation. Sustainability is a big thing of what we do, and GeneStream is our genetic services. We provide educational material and tools to our customers and to our internal staff to educate them in their journey. As you can tell, genomics is becoming a very, very complex thing. And when you have your salesperson that were graduated 10, 20 years before genomics became a thing, it's very difficult for them to keep up with those new terminologies. So that's where our duty is actually to give them as many tools as we can to allow them actually to sell genomics and genetic progress. This is my team. I have one Iowa State former here from Jack's team. He's doing great, by the way. I mean, I have 12 people, that most of them in animal breeding and genetics. That this is my core team, that we are focusing on R&D, basically in applying or developing new genomics tools to our company, so we continue to advance genomics progress. Back to where I started, where, I cannot imagine when I first joined the lab in January 1999, me, my wife, and a little baby was 6 months old. I will be talking about genomics to this level as I'm talking today. So thank you, Sue. It's been a pleasure. It's been an honor, and it's an endless learning process from day one I joined your team until today. Thank you. Thank you, everyone.
Thanks, Joseph. It's always fun to hear about the other species. Mohammed, question.
Yeah, thanks for the presentation. I'm looking at it from a testing point of view, where you kind of showed from how you estimate your ABVs and two processes where you could get the amniotic fluid, and then you can isolate the embryonic cells, and then you go to the genotyping from there. Or you can wait after calving and you can take a tissue sample down the line and then do pretty much the same thing. So from a cattle standpoint, which is the most standard? Is it the amniotic fluid or you wait after calving? And also, what's the cost implication of the two?
Yeah, good question. So the question was about what approach do we actually use today in our program? Do we do amniotic fluid, or we do embryos, or we wait until the animal is born? If I understood you correctly, we do all the above, basically. It depends, right? Because we have really very advanced genetics, right? And every animal is born is really heavy value, right? So it's not always like we want to limit the selection early on, right? It's sometimes makes sense just to produce to keep whatever is born out of that litter now of full SIP animals, right? So sometimes we do we take everything to term and we test after birth, and sometimes we do amnio, and sometimes we do in vitro. It's just about managing resources. That's all. I mean, it's not about genetic progress, right? We still, we are very focused on genetic gain and genetic progress, but it's about just managing resources, right? What is available at that time.
Thank you for the nice presentation about a lot of genomics. And my question is related to the previous speaker, that what is the implication on phenotyping on when you drive this so fast? What kind of resource do you need, and how do you organize it?
Yeah, good question. So the question is about phenotyping, and I didn't mention that at all. So yes, that's another side of everything we do. As I said, like in the first part here with the genetic and genomic, that's where actually we spend time in collecting phenotypes, analyzing those phenotypes, and bringing those to be testing. We do tons of tracking or measuring new traits. My baby has been, since I joined the company, is the feed efficiency project, what we call an eco feed today. It became a product. So we have spent millions of dollars just measuring individual feed intake on cows and heifers and beef animals, those phenotypes feeding into a breeding or genetic evaluation, and we calculate genomic breeding value of all animals, and we publish that in our catalog of all animals that for all customers that genotype with us. So that's one example of kind of deep phenotyping that wasn't cheap, and we spent time on that. We have a program to measure. So we have another branch of the company. We're focusing on technology, on sensors technology. So produce boluses that actually measure body temperature and movement, etc., that goes into the rumen. And we have body temperature on those animals every second of their life. And that is feeding into another program. We actually try to develop systems or breeding values for heat tolerance for those animals, because I have their production, I have their body temperature, inner body temperature, and I have the ambient temperature at that minute. Right? Some of those animals in Texas, I'm in Texas, by the way, that's where my accent is from, by the way. So we don't have a shortage of heat stress days.
Sorry. Thank you for a fascinating presentation. I think you gave a master class on the possibilities of genomics and genetic techniques, and I think you deserve 2 hours in the program to go through them in more detail. But my question is, my experience in poultry, both in layers and broilers, is that there is a lot of scrutiny into the use of some of these technologies, and we have to write the letters or statements indicating what kind of biotechnology and reproduction technology we're using in terms of, for instance, cloning or production of GMOs, etc. What level of scrutiny or the public reaction do you have when you describe some of these methodologies, or is none in the case of cattle? Thank you.
Yeah, I mean, that's also a good question. So the question is about the public acceptance of those kind of reproduction technologies implemented in dairy. I cannot answer that easily, right? It's a complex question, and it varies from country to country, and the regulations are vary from country to country. Even within the US, different states allowing you different things, right? So like some states, a technician in the farm cannot do the amniotic testing, but other states, it's okay. You have to be a vet to get an amniotic fluid sample out of those cows, right? For example, right? There's a lot of regulations that we adhere to, of course. And in terms of the public acceptance, yeah, it is. This is not a subject that comes up a lot in public sessions, right? To keep it short.
I was told what technology do you use for sequencing, and then would you just open the door a little bit on your computer facilities?
So in terms of sequencing, we just Illumina what is the name of Nova 6000, something like that. I can't remember something. It's a monster of machine. In terms of our computing powers, we do everything in-house. Basically, we don't do cloud computing or anything like that. It's just a choice of the company. We continue to buy servers, we continue to buy CPUs, and we continue to buy, terabytes of storage. I already filled out 500 terabytes of sequence already. We just bought another 1 petabyte of new storage, right? So I know that cannot continue forever, but we try to find the right balance. Yes.
Joseph, thanks very much. Very interesting. Part of the reason you can do some of these things is a cow's worth more than a pig is worth more than a chicken. So in terms of some of these technologies going downhill to the other species, how do you see that happening relative to in reducing costs and making it possible?
Good question. Yeah, I definitely, as I said, like in my introduction, that the bovine is provided the study case for applying genomics. And if it wasn't for bovine, we wouldn't be doing genomics in pigs and chickens, honestly speaking, because they showed that how successful that has been. Now, in some cases, I make some of my colleagues angry when I say, like, in some species, like in certain traits in broilers, for example, application of genomics doesn't make any sense. I hope I'm not offending anyone here, right? And depends on the trait and depends on the directions you want to go. So it is depending on what traits you have and what direction you really want to take your genetic program. And in some cases, it really doesn't make any sense to do genomics. You have species that have low value and short generation interval, and you're looking for only body weight. Why not just keep producing the hell of them, right? And selecting the one that actually exceed, the genetic variance you were expecting, and produce from that one.
Hey, I get to be the bad guy to cut this great discussion. So thank you, Joseph.
Thank you.